Good morning, ladies and gentlemen. My name is Nick Riker, Director of Policy and Research at the MS Society. Before I start, we have had a fall in the room, and I just want to check that it's appropriate for us to continue rather than wait. Yeah, okay, good. Thank you. This morning is going to be a fascinating discussion. Hopefully you would know that over a number of years now the MS Society has been investing pretty heavily in myelin repair, crucial area in terms of the breakthroughs we're all looking for in MS research. Some of you will have heard the conversation yesterday where we talked about one day arriving at a state where we'll have combination therapy. Some of the therapies that people today are able to benefit from, coupled with therapies around myelin repair and neuroprotection, that together could form really effective combination therapy for MS. So MS could then be managed a bit like HIV is today, much more effective, where people don't have to live in so much fear of what might happen to them, that they, with hope, wouldn't then be experiencing the range of symptoms that people typically experience today. So that's what we're really gunning for. Crucial to that is the work that we're doing around myelin repair. We've invested very much in work being done at Cambridge primarily, but also Edinburgh. They've been working very closely together to push this forward. And I'm delighted to say that today we have, I would say, one of the world's leaders in terms of myelin repair, Robin Franklin. Robin has a really interesting background, which I won't necessarily go into now. He may well tell us, or you could ask him. Fascinating as to how people form their medical careers and the paths they go through to get to where they are. Robin is Professor of Stem Cell Medicine at Cambridge. He works in an institute funded by the Wellcome Trust and the Medical Research Council. He's also head of translational science. And very importantly for us, Robin is director of the Cambridge Centre for Myelin Repair, which is funded by the MS Society. So Robin, if you'd like to talk to us, we'll then have plenty of time for questions and answers. Thank you.
Nick, thank you very much for that kind introduction, and thank you very much for coming along this morning to hear what I have to tell you about the work that we've been doing in my centre in Cambridge and also in collaboration with the Edinburgh Centre, which is co-directed by Professor Charles French-Constant and Professor Siddharthan Chandran. So, this is what is in store for us over the next 45 minutes or so. So I'm going to try and provide some answers to the following 4 questions. Firstly, describe to you what myelin is, and more importantly, what it does. Secondly, describe exactly what we mean by the term myelin repair, or more accurately, the term remyelination. These are synonyms, but and I'll explain what they mean in just a moment. Then thirdly, why myelin repair is a very important objective in this sort of comprehensive, multi-component strategy towards for MS therapy. And then finally, and perhaps of most interest to most of you, where we're up to in our joint centres in identifying ways that we can therapeutically make this regenerative process more efficient in the clinical context. Okay, so let's kick off with the first of these questions, myelin, what it is and what it does. So here is a diagram which I've used to introduce my talk for many years, and the reason I've used it often is because it's very simple and a very, I think, clear way of describing the key components of the story that is going to unfold. So what you see here are the crucial cellular constituents of the central nervous system. The central nervous system is the term we use to describe both the brain and the spinal cord. So what you see over on the left of this diagram are 3 grey circles, which are nerve cells or neurons. Nerve cells and neurons have axons or nerve fibers, which head off some considerable distances often to connect with other nerve cells or neurons, and thereby construct this extraordinarily complex circuitry, which is characteristic of the human brain and spinal cord. In fact, it's probably the case that the wiring of the human brain is the most complex structure in the known universe. Not that we know everything in the known universe, but, well, no, we do know everything in the known universe, but not in the universe. Anyway. So the other cell type you see there is the green octopus-like cell at the top of the image. And this is a cell that we call the oligodendrocyte. There's no simpler term for it. It's the oligodendrocyte. And the oligodendrocyte is responsible for making the myelin sheath, which surrounds and wraps the nerve fiber or axon. And that's perhaps illustrated more clearly in the following diagram. So here in the middle, you see the oligodendrocyte, a cutaway diagram of that important and crucial cell. And you can see that there is a central component called the cell body with a nucleus within the middle, which is a dark brown structure. And coming off from the oligodendrocyte cell body are short processes which end in large sheets of membrane which wrap Swiss roll style around the axon. And that constitutes the myelin sheath around the nerve fiber. And you can see that a single oligodendrocyte can make many individual myelin sheaths. In fact, a single oligodendrocyte can make up to 80 separate myelin sheaths surrounding 80 nearby nerve fibers. Here is what the myelin sheath actually looks like when viewed by electron microscopy. So this is looking at the myelin sheath of a rodent. This is many tens of thousands of times magnification. And here you're looking at a nerve fiber cut in what we call transverse sections. So this is like slicing French bread. So in the middle, the pale area is the nerve fiber axon, and the dark staining structure around the outside of the axon is the myelin sheath. And you can see these concentric rings of dark staining, which are formed by the wrapping of the membrane of the oligodendrocyte around the nerve fiber to form the myelin sheath. So what does the myelin sheath do? Well, there are two key functions of the myelin sheath, and it's important to understand these to understand why symptoms arise when the myelin sheath is lost and damaged. The first function of the myelin sheath is one that we've known about for many years, and that is the function of conferring on the nerve fiber the ability to conduct electrical impulses much more rapidly than would otherwise be the case. So the way that nerve cells communicate with each other is by way of electrical signals which form a continuous conducted impulse down the nerve fiber. And as a result of the myelin sheath, for reasons that we don't need to go into any detail, that process is made very much quicker than would otherwise be the case. So because of the myelin sheath, the action potentials, these electrical signals, can whiz down the nerve fiber with some speed. And if you do the maths, you can figure out that in terms of something that's perhaps easy to understand, in the absence of a myelin sheath, the electrical signals would travel at the speed of a tortoise, but in the presence of the myelin sheath, the electrical signals travel at the speed of a fast-running cheetah. And those of you who know something about the animal kingdom, which I assume is all of you, will know that cheetahs are very much quicker animals than tortoises. So you can see that in the absence of the myelin sheath, the electrical signals are proceeding very slowly. In the presence of the myelin sheath, the electrical signals proceed very rapidly. Now, the second important function, and perhaps in some ways the more important function when one thinks of the long-term implications of the disease, the second important function of the myelin sheath is that it protects and in some way nourishes the underlying nerve fiber. And here the best analogy is probably the lagging around the hot water pipes in your house. So in the presence of the lagging, the hot water pipe is protected, so you can have a cold spell and you know that your hot water pipes are going to remain intact. But if you don't have lagging around the hot water pipes and you have a cold spell, then the hot water pipes can snap and break. And just as the breaking of nerve fibers in the central nervous system, those hot water pipes won't spontaneously regenerate. In fact, in the central nervous system, if a nerve fiber breaks, it's all but impossible to get that nerve fiber to regrow. So preventing the loss and damage of a nerve fiber is a crucially important thing to try and achieve in any clinical situation where there is potential vulnerability of the nerve fiber in the central nervous system. So those are the two key functions of myelin in the central nervous system. One is to allow electrical signals to proceed much more rapidly, and secondly, the myelin sheath protects the underlying axon, preventing it from being damaged or, frankly, snapping. So that's the key constituents of the central nervous system, the nerve fibers and their axons, myelinated by an oligodendrocyte, which makes myelin sheaths around the nerve fibers. Now, MS is a disease characterized by a disturbance of the immune system, such that the immune system gets itself confused and starts to imagine that components of the central nervous system shouldn't be there. And the specific component that is targeted in multiple sclerosis is the oligodendrocyte and the myelin sheath that it makes. So you have an immune system which goes awry for reasons that are still far from clear. It kills the oligodendrocyte. As a result of the death of the oligodendrocyte, the myelin sheath comes off the nerve fiber, and the nerve fiber is no longer myelinated. And of course, that'll have immediate implications for the ability of that nerve fiber to conduct electrical signals, and will eventually have implications in terms of the health of that axon, as I'll come on to in just a moment. So, in the absence of the myelin sheath, the action potentials now proceed at the speed of a tortoise rather than the speed of a cheetah. Okay, so let's now get on to the second of the questions that I posed at the beginning of this talk. What do we mean by the term myelin repair, or more accurately, remyelination? So, when oligodendrocytes are lost in the central nervous system, as occurs in an acute episode of the disease that we're discussing today, there is a spontaneous regenerative process that takes place. So the central nervous system, in all adult mammals, including adult humans, has the capacity to regenerate new oligodendrocytes. And this is a very positive and optimistic thing, because it contrasts very sharply with the consequences of the loss of the nerve cell. So if you lose the nerve cells, the 3 cells that you see over on the left, as is the case in the classic neurodegenerative diseases such as Parkinson's or Alzheimer's or Huntington's disease, the adult human brain has pretty much no capacity to generate new neurons. So if you lose neurons, they're gone forever. However, if you lose oligodendrocytes, you can regenerate oligodendrocytes with some efficiency, subject to various variables that I'll come on to shortly. So the regeneration of new oligodendrocytes is the process of myelin repair or remyelination. And those newly generated oligodendrocytes put new myelin sheaths around the nerve fiber, and so the whole architecture of the tissue is restored to what it was originally. So this is a true regenerative process. And this is really why I actually prefer the term remyelination than myelin repair. Myelin repair gives some sort of sense that there's holes in the myelin which you patch up. That's not what happens at all. The myelin sheath disappears in its entirety and it is replaced in its entirety. So it's a true regenerative process rather than a patching up repair process. So how does remyelination take place? How is it that the brain is able to regenerate new oligodendrocytes in the face of their loss? So now here's a somewhat more complicated diagram of the key constituents of the central nervous system. So running left to right are the green lines which constitute the axons or nerve fibers, and they'll be attached to a neuronal cell body at some point over to the left or right. And those nerve fibers are myelinated, as we've already discussed, by oligodendrocytes, which are the yellow cells that you see with processes going out top and bottom, attaching to the myelin sheaths that are surrounding the nerve fibers. Now, the other cells that I've included in this diagram are the blue cells with little processes sticking out to left and right. And these are the stem cells of the adult brain. Technically, we often call these cells oligodendrocyte progenitor cells, or OPCs, but they are, in effect, the stem cells of the adult brain. And it turns out that the adult human brain, the adult mammal brain, is full of these stem cells. They are a very abundant population. Probably about 6 to 8% of all the cells in your brain are these brain stem cells, widely and evenly distributed throughout the whole of the brain and spinal cord. That's most easily seen if you look in a laboratory species like a rodent, a rat, or actually in the image I'm just going to show you a mouse, where we can actually identify individual brain stem cells. So what you see here is a section through the brain of an adult mouse, cut in this direction, so we're going through the middle of the brain. And the reason this image looks slightly purple, and if you're in the front few rows you might see that there are individual dots, is because each of those individual dots that you see within this adult mouse brain is a brain stem cell. So you can see that they're very abundant, widely distributed throughout the whole of the mouse brain, and the same would be true if you followed on down the spinal cord. And the same would be true that if you looked in a human brain. So these stem cells are very abundant, and that's an important point for reasons, again, I will come on to shortly. So, how do these brain stem cells contribute to the process of remyelination? So let's imagine that within this area of tissue, where we have myelinated axons running left to right, an episode of demyelination takes place. So demyelination is the process which occurs when the oligodendrocytes are killed and the myelin sheaths come off the axon, leaving them demyelinated. So we've killed the oligodendrocytes, and as a result of killing the oligodendrocytes, the myelin sheaths degenerate, and the axons become demyelinated and exposed. And of course, as I've already mentioned, that will have immediate consequences for the ability of that nerve fiber to conduct electrical impulses efficiently. Now, in response to that damage, that injury occurring in the brain and spinal cord, the whole milieu of that tissue changes in such a way that the stem cells that are present in the vicinity of the injury are stimulated to respond. So the first thing that happens after you lose oligodendrocytes is that the brain stem cells around the area of damage recognize that something has gone wrong. They change their whole state of being and start to make copies of themselves. In other words, they proliferate, and they also start to move around, they migrate. And so what happens is that that area of damage fills up with recruited brain stem cells. So they respond to signals that are generated by the damaged tissue. They become activated. They don't actually throb like this, but that's just illustrative of their change in state. And in that changed state, they make copies of themselves that move around, and they fill up the area of damage. And that's the recruitment phase of remyelination. So very quickly after you lose oligodendrocytes, the area of damage is filled up with brain stem cells. This is an illustration from an experimental animal where we've been able to genetically label the stem cells so that they fluoresce green. And this is a very, very small component of the spinal cord of an adult mouse where we've killed the oligodendrocytes. And shortly after the loss of the oligodendrocytes, it's such a small area that the mouse is completely oblivious to the fact that we've killed a few oligodendrocytes at some point in its spinal cord. And shortly after the loss of those oligodendrocytes, that area of damage fills up with these stem cells. So you see this area of damage now chock-a-block full of green stem cells. And these sort of models are extremely useful to us as experimental scientists studying the process of remyelination, because we can now see the stem cells and we can study their behavior in some detail and the factors which regulate and control their behavior. So the final phase of the regenerative process of remyelination is when the recruited stem cells become the missing cells. And the missing cells here are, of course, the oligodendrocyte. The process of a stem cell becoming a very specialized type of cell, such as an oligodendrocyte, is the process that we call in stem cell biology differentiation. So the second and final and critically important phase of remyelination is when the recruited stem cells become oligodendrocytes that grab hold of the demyelinated axons and reinvest them with new myelin sheaths. And at the end of that process, again, the architecture of the tissue is restored to normality. So the axons are now surrounded by myelin sheaths, which will allow them to conduct by this rapid mechanism of saltatory conduction, the speedy conduction of impulses along the length of the nerve fiber, and will also help protect the nerve fiber. So that, in a nutshell, is how remyelination takes place. Of course, the mechanisms driving this are very much more complicated than I've conveyed in these diagrams, and we'll come on to some of those mechanisms shortly. So the third question is, why is myelin repair such an important process? And given what I've said so far about the function of myelin, you can begin to anticipate what the answer to that question might be. So let me just step back from this and give you some important context to why myelin repair is such an important process. So represented here is the inescapable process which affects all of us of aging. We are all aging all the time. So we're now older than we were when I started this talk, and you may feel that you're much older than you were when I started this talk. So, multiple sclerosis is a long-term disease. Commonly, it occurs in individuals in early adulthood. And as you know, the life expectancy of people with the disease is not shortened by that very much compared to the normal population. So this is a disease that people will live with for several decades. So this is a disease that people commonly have throughout their adult life. So the process of aging is likely to be pertinent to how the disease unfolds as you move through adult life. And it transpires, and is perhaps not surprising, that perhaps the most important determinant of any regenerative process in any tissue is the process of aging. So as we get older, our ability to regenerate tissue decreases. And in some sense, that's why we age, because we're not able to regenerate tissues as efficiently as we were when we were younger. This is illustrated in this first data slide of my presentation, which is taken from a paper published over 40 years ago. And it's a classic paper which looked at the effects of aging on a very simple regenerative process of wound healing. So this is a breach in the skin, a wound that then has to heal. And between the ages of 20 and 40, the rate of healing of that wound is halved. So it takes twice as long to heal a skin wound between the ages of 20 and 40. And this is a progressive process. So if you looked again at 60 and 80 and so forth, the rate of wound healing would be decreasing as you age. So this is really illustrative of this very important concept that aging affects the efficiency of all regenerative processes. And it turns out that you could replace any tissue you like here, and the phenomenon would be exactly the same. So you could put in liver, you could put in the bone marrow, you could put in any tissue you like, you could put in bone here, and it would be profoundly affected by aging. So here is some other diagrams illustrating this concept of the effects of aging on regeneration, looking at the ability of bone to regenerate. This is a young adult rat of about 3 months of age, and an experimental fracture has been made at this point here. And 80 days after fracture, it's almost impossible to tell where that fracture took place because the bone is healed completely. But in an old adult rat that's about 24 months of age, at 80 days, it's still very clear, if you peer closely, but it is pretty clear, that bone is still not healed. And that, of course, is why skiing is a pretty reasonable sport for somebody in their early adult life to engage in, because if they break their leg, it's going to heal pretty quickly. But if you're in your 60s, 70s, and 80s and you go skiing and you break your leg, then it's going to take a lot longer to heal. So this phenomenon of aging affecting the efficiency of regenerative processes occurs in all tissues in the body and absolutely applies to the efficiency of regeneration of myelin sheaths. So as we get older, as we progress through adult life, with the disease, our ability to regenerate new oligodendrocytes is declining simply as a function of the clock ticking. It's got stuck. Here we go. Okay, so the implications of that is that early on in the disease, once this process has occurred, so you have an episode of inflammatory demyelination taking place at some point in the central nervous system, if you're a young adult, then the chances are you'll regenerate oligodendrocytes pretty quickly and pretty efficiently. But as you progress through the disease, as you age, the efficiency of this process is declining, and it can become so slow that, to all intents and purposes, it appears to have failed. So remyelination could become so inefficient that, de facto, the remyelination has more or less ground to a halt. Now, if demyelination occurs and there is no regeneration of the myelin sheath, the axon is then left exposed for longer than is good for it. So not only does that axon now lose the ability to conduct electrical impulses efficiently, but because of the second function of the myelin sheath, of maintaining the health of the underlying axon, in the absence of the myelin sheath, that axon is very vulnerable and liable to undergo degeneration itself. And as I mentioned earlier, if you lose axons, nerve fibers in the central nervous system, there is precious little by way of spontaneous regeneration. So this is an irreversible change. And sometimes, in fact, the whole nerve cell can die, not just the nerve fiber, but the whole nerve cell. And again, as I mentioned earlier, if you lose nerve cells, they're not spontaneously regenerated. So if you get to this point where there is no restitution of the protective myelin sheath, the nerve fiber will degenerate, and because that's irreversible, the accumulation of loss of nerve fibers leads to the progression of the disease. And so what is really happening in the secondary progressive phase of the disease, in fact, it's happening already in the early relapsing remitting phase, but certainly in the progressive phase of the disease, there is the driver of that is the cumulative loss of these irreplaceable neuronal elements of the central nervous system. And that's why putting myelin sheaths back on axons is a critically important way of slowing or arresting the progressive phase of disease. And our efforts in Cambridge and Edinburgh over the last 8 years or so have really been focused on this ultimate endpoint of coming up with some strategy for trying to address the, at present, untreatable progressive phase of MS. So clearly, there is an important therapeutic, urgent, I would say, need for making sure that when this process occurs in the central nervous system, you always go down the regenerative protective route of reconstituting the tissue and putting the protective myelin sheath back on the nerve fiber, and avoiding going down this degenerative route where remyelination has failed, and degenerating because remyelination has failed. So let's now think about how a regenerative therapy fits into a broad strategy for treating multiple sclerosis. Now, clearly, if one was thinking about this as a conceptual thought experiment, the first thing you'd want to do is to try and stop the damage occurring in the first instance. Clearly, you'd want to try and intervene at this point here to stop demyelination taking place. Now, as I've mentioned to you, and you'll have probably heard from others on many occasions, the demyelination is occurring because the immune system goes awry and starts to kill oligodendrocytes and damage the myelin sheath. So clearly something that changes or modulates or suppresses the immune system will help in preventing this demyelination occurring in the first instance. So the first strategy of stopping damage is the whole area of immunomodulation, the immunotherapies. And pretty well everything that is currently available that you may be on or may want to be on or might be on at some point in the future, all of the drugs that are currently available, the disease-modifying drugs, are really aimed at modulating the immune system such as to stop the damage occurring in the first instance. Now, ultimately, it may well be the case that those therapy interventions are so effective that we can completely shut down the disease from the outset. That, of course, would be a very that would be an objective that one would hope to reach at some point in the future. But here and now, it's naive to think that we're going to be able to completely suppress that damage from taking place. So it will be the case that some of the some disease process will escape the disease-modifying interventions, and demyelination will take place. And at this point, you really want to have a regenerative therapy, which makes sure that you go down the regenerative pathway and not down the degenerative pathway. And by implication, what I said earlier, the further you on in the disease, the greater the urgency and the need for regenerative therapy, because your own natural healing processes will be becoming tired as a result of aging. So this has really been the focus of the two centres in Cambridge and Edinburgh over a number of years, to see whether we can come up with ways by which you can promote myelin regeneration in those situations where we haven't been able to completely suppress the disease process by the currently available disease-modifying drugs. And this is now, I think it's true to say we were sort of pioneers in this area and have been for a number of years. And the MS Society, of course, has been, in that sense, also a pioneer in their investments in Cambridge and Edinburgh. But is now, I think, universally recognized internationally as the major urgent objective in new therapies, new radical therapies for this disease. So then the last question, how can we do this? What approaches can we take to try and make sure that we put myelin sheaths back on axons after demyelination has taken place? Well, the classical way of thinking of regenerative medicines, the approach that you read about in the daily press is the concept of cell transplantation. For many people, this is the top of the list of the ways that you can regenerate tissue. You transplant a cell into the damaged area, and that transplanted cell replaces the cells that are missing, and you're back to where you were, healed. And for many people, the best cell to transplant would be a stem cell, with the proviso that I'll add that the term stem cell actually embraces a whole range of different cell types. There is not a single entity of a stem cell. So one has to be very wary when you read reports of the efficacy of stem cells in the paper, firstly, whether it's true or not, but secondly, precisely what type of stem cell is being talked about. There is not a single entity of a stem cell. But it would not be unreasonable to think that in a demyelinating disease, you might be able to transplant brain stem cells that we know are capable of making the oligodendrocytes into an area of demyelination in the brain, and those transplanted cells would replace the missing oligodendrocytes. Well, that conceptually is a very straightforward approach to myelin regeneration. Well, the first thing is, where are we going to get these cells from? So that's not a trivial question, by the way. One possibility is that you could actually grow these cells from the adult brain. So you could take a bit of brain from a patient, identify these cells, grow them in tissue culture, make lots of them, and then transplant them back into the brain. And given what I said to you earlier, that there are an awful lot of these cells in the brain, on the face of it, that seems not to be an unreasonable approach. And it seemed to be a not unreasonable approach when we first entertained this idea about 20 years ago. Transpires that it's extraordinarily difficult to grow these progenitor cells from the adult brain for reasons that are still not really understood or been addressed. In the last few years, alternative ways of growing stem cells have emerged. So you may have come across the concept of the iPS stem cell. This is the work that Yamanaka and Gurdon won the Nobel Prize for last year. And this is the idea that you can take any cell in the body, so you could take some cells from the skin, and that's a very trivial way of obtaining cells, and by a relatively short number of steps, in fact, a surprisingly short number of steps, you can make any cell in the body into an embryonic stem cell. Embryonic stem cell has the wherewithal of to become every cell in the body, and so you can go from the embryonic stem cell towards the cell that you want. So you can take any cell in the body, make into embryonic stem cell, and then differentiate that embryonic stem cell into, for example, a brain stem cell that has the capability of becoming oligodendrocyte. And that technology is rapidly becoming available. So there are now a number of publications where people have described methodologies for obtaining human oligodendrocyte or brain stem cells from something as trivial as a skin cell. But there is a second problem in addition to where you would obtain the cells from, and that is the feature of multiple sclerosis, in that this is not a disease where there is a single area of damage in the brain that occurs as a one-off event. Instead, as we know, the damage within the MS brain occurs in many points at different times. So transplantation for multiple sclerosis would be very different from cell therapy approaches, for example, to spinal cord injury, which some of you might have come across in the press, where you have a single episode of damage occurring at a single point in the spinal cord. So you put cells in there, and that's relatively straightforward. Same would be true for Parkinson's. There's a single area within the brain where you lose neurons, and so you transplant cells into that single area within the brain. That is not true in multiple sclerosis. The damage is occurring at different points throughout the brain and spinal cord. And so that makes it a challenge to think how you would transplant into all of those areas at different times during the disease course. And that is not a trivial challenge by any stretch of the imagination. And it is largely for that reason, actually, that many of us have become more attracted to a second approach to promoting remyelination in the MS brain. But before I get onto that, I just want to clarify something which I think causes a lot of confusion in the community. So there are stem cell therapies available at present and being trialed in various countries for multiple sclerosis. In all instances, I think I'm right in saying the stem cells that have been used here are stem cells obtained from the bone marrow. Very often they're a type of stem cell called a mesenchymal stem cell. Not always, they're sometimes hematopoietic stem cells, but generically bone marrow stem cells. Now, I think it's very important to understand because there is this sort of zeitgeist and popular conception at the moment that stem cell transplantation is always a regenerative strategy. That is not the case in MS when you transplant bone marrow stem cells. So bone marrow stem cells are not directly regenerative in MS. I thought that was my cue to shut up, but right, so let me just explain that now in the context of the two approaches that I described to you for treating MS. So you remember I said there were two approaches. One is to stop the damage from taking place, and that's to modulate the immune system, and the other is to promote the regenerative process. Bone marrow stem cell transplantation is really targeting this. It's stopping the immune, it's modulating the immune system, which is causing the damage. So that we know that bone marrow stem cells, and particularly mesenchymal stem cells, are very efficient at changing immune systems, changing the immune response. Exactly how they do that is still a bit of a mystery, but we know they're very efficient at doing that. So bone marrow stem cell transplantation is not a regenerative therapy, it's an immunomodulatory therapy. So it is in the same category of treatment as all of the drugs that you know that are currently prescribed and used for the treatment of the disease. They are not directly regenerative therapies. So you do not go to get a bone marrow stem cell transplantation therapy with a view directly to being regenerated. That's not how bone marrow stem cells are working, and I hope that's clear. It may be the case that in modulating the immune system, they change the environment in such a way that this remyelination process is more likely to take place, and that's an intriguing possibility for which some evidence exists. But it is not the case that these bone marrow stem cells will generate new oligodendrocytes directly as a regenerative process. Okay, so that's really a summary of transplantation approaches to curing or remyelinating in multiple sclerosis. Brain stem cells will make oligodendrocytes, but how you get them to all the different areas in the brain where they're needed is a difficult challenge. And bone marrow stem cells, when they're transplanted, are not directly regenerative. That's why some of us have been entertaining this alternative approach to promoting remyelination. That is based on the fact, which I've already introduced to you, that the brain is full of stem cells that are capable of becoming oligodendrocytes. So we know that these stem cells can become oligodendrocytes. And we know that they do that very efficiently in young adults. And we also showed in a paper that received some attention about 80 or so months ago that even the aging stem cell is still capable of making an oligodendrocyte. So it ought to be the case that you can do something to the stem cells that are already present in your brain to make them regenerate oligodendrocytes more efficiently. So instead of transplanting stem cells, you target the stem cells that are already present in the brain. This is sometimes called the endogenous approach to remyelination because you're targeting the endogenous stem cells already present in the brain. And that is going to be, in all probability, a pharmacological approach. Instead of a transplantation of cell approach, instead a drug which somehow changes the behavior of these stem cells, making them become oligodendrocytes more efficiently. So this slide here just summarizes the conceptual approach that we've taken over a number of years to try and see whether we can come up with drugs that make stem cells, make oligodendrocytes more efficiently, and would therefore be the basis of regenerative medicines. Now, if your car breaks down, you are in no position to fix it unless you understand how the engine works. So a priori to fixing a broken car is you have to understand how the engine works. And if you understand how the engine works, then you can figure out what's gone wrong when it breaks down. And if you know what's gone wrong when it breaks down, then you can repair the car. So all people that come out to fix your car understand how the engine works. I'm certainly not in that category. But that's pretty much the approach that we have decided to adopt in coming up with ways of promoting or fixing myelin regeneration, myelin repair. First of all, we have to understand how remyelination works when it works efficiently. So how does the car engine work when it works normally? How does remyelination work when it works efficiently? If we understand that process, we can understand why it goes wrong, and if we understand why it goes wrong, then we can fix it. And that is why much of the time has been spent, much of the investment has been spent from the MS Society in really trying to get to grips with the fundamental biology of how remyelination takes place. So the diagrams that I showed you earlier on showing how remyelination occurs, where you get stem cells that get recruit and differentiate, that was crucial information to understanding this process. And a lot of that understanding has come from work that we've done with MS Society investment. Now, it turns out that as you get older, the stage of remyelination which is most seriously impaired by the process of aging is not the abundance of stem cells. The number of stem cells in your brain stays about the same regardless of your age, nor is it their ability to be recruited, but it's the final stage of remyelination where the recruited stem cells have to become new oligodendrocytes. So commonly, if you look in areas of the MS brain where remyelination hasn't taken place, it looks like this. So you see demyelinated axons, and you see plenty of these stem cells, but they haven't yet been able to become oligodendrocytes. And it's for that reason that we've spent a lot of time in the two centers in trying to understand the mechanisms, the machinery by which a stem cell is able to engage an axon, become an oligodendrocyte. So what are the intrinsic and extrinsic processes, in other words, the processes within the cell and the processes around the cell that are making that stem cell become an oligodendrocyte? This turns out to be an extraordinarily complicated bit of biology. There are multiple mechanisms working interactively with each other in different degrees of importance. But we have been, I think, rather successful over the last few years, again as a result of the investment the MS Society have made, in identifying a whole host of critical processes which are involved in stem cells becoming oligodendrocytes, and those are listed here. Needless to say, these processes are related to pathways or molecules and therefore have the somewhat abstruse names that typify biological science. But these pathways or molecules are listed on the left here. They've, for the most part, well, pretty much always been published in very high-impact journals. And some of these processes or pathways or molecules are processes which make stem cells become an oligodendrocyte, and some of them are processes which stop cells becoming an oligodendrocyte. And if they're responsible for making a stem cell become an oligodendrocyte, they're blue, and if they're responsible for stopping a stem cell becoming an oligodendrocyte, they're in red. So if it's a process which makes a stem cell become an oligodendrocyte, then you design a drug which replicates that. If it's a process which stops a stem cell becoming an oligodendrocyte, you design a drug which inhibits that inhibitory. So two inhibitions cause a positive. So this first target was the first one we published. This was a collaborative work between my lab in Cambridge and Charles French-Constant's lab in Edinburgh. The Wnt pathway was a collaboration between my lab in Cambridge and some colleagues at University of California, San Francisco. Endothelin-2 and activin-A were both targets or mechanisms which were joint ventures between Cambridge and Edinburgh. PDE4 was a collaboration between my lab and a colleague in Cambridge, Mark Cottrell, I'll mention in just a moment. And this is a paper which is going to come out shortly where we've identified another mechanism by which you can promote remyelination, which has been done in collaboration, again, with colleagues at University of California, San Francisco. Let me just highlight two of these that have been published within the last 6 months. The first of these is a molecule called activin-A, which actually turns out is made by inflammatory cells which arrive in areas of damage. So it turns out that not all inflammatory cells are necessarily bad. There are types of inflammatory cell which turn out to be quite good and rather important for regeneration to take place. And those good inflammatory cells produce a range of molecules which help the regeneration of myelin take place. And one of those molecules is activin. And this was discovered by Veronica Miron, who is a postdoc working jointly between my lab and Charles's lab, and now is running her own laboratory at the University of Edinburgh. This is very early days in translating this particular discovery into a therapy. There's still a long road before that takes place. but it's very important to have identified these starting points in that sort of translational process. And the second of these, the phosphodiesterase inhibitors, this was work that was led by Mark Cotter, who was an ex-student of mine, who now runs his own laboratory in Cambridge, and we published this jointly last year. And these are interesting, exciting stories which will unfold over the next few years, and we hope eventually lead to new therapies. Now, there is a pipeline, a process from going from a scientific discovery in the laboratory to a clinical intervention. And that pipeline can be long or short, depending on what's available, what's known, what the type of discovery is in the first instance. So the first of the discoveries that we made and published a few years ago was this compound here, RXR gamma. And we're just going to tell you a little bit more about that, because that is the one that is furthest along this pipeline from being a laboratory discovery to a clinical intervention. So RXR gamma is a very interesting molecule that is rather complicated. Part of the molecule can receive drugs and compounds coming into it and can attach to that molecule, so it acts as a receptor. And another part of the molecule will actually engage in the nucleus and change the whole behavior of the cell. So these are really rather interesting and important molecules, and I can tell you a lot more about those should you wish, but that's the essence of what these molecules do. So RXR gamma is expressed within brain stem cells, and if you activate this molecule, the stem cell becomes an oligodendrocyte much more efficiently. So what we showed in the paper we published a few years back was if you take old rats, so these are old rats that when you demyelinate them, they're going to remyelinate very slowly, and you give those rats a drug or a compound which activates that molecule RXR gamma, you can make the remyelination much more efficient. And what you have to grasp in this figure here is that if it's a pink circle, the nerve fiber has been regenerated with new myelin. If it's a gray circle, it hasn't. So this is the control old animal, which has just been given saline, salt water. Very few of the axons have new myelin sheaths and are pink. We've made them pink. That's sort of they're not actually pink. Very few of them are pink, but if you give a molecule which activates this RXR receptor, then many more of them are remyelinated. And this actually was quite a dramatic discovery. So this was actually the first time we, or anybody for that matter, had made an old animal make myelin sheaths more efficiently by giving a single molecule. So you can see here very clearly that we've promoted remyelination. There are more pink fibers on the right than there are on the left. So RXR gamma is a very important target for making stem cells into oligodendrocytes. Are there any drugs which target this receptor that might be available as a regenerative medicine in MS? Well, as I said, the rate at which you move along the pipeline varies from what's available and what's known and what the target is and so forth. Actually, for the RXRs, we can sort of fast forward along the pipeline reasonably quickly because it transpires that there is already in existence a drug developed and licensed for a completely different indication which targets this particular receptor, and that is a drug called bexarotene. So, bexarotene activates RXR much in the same way as the molecule that we used experimentally in the previous slide I showed you. So would it be possible to use bexarotene as a regenerative medicine in MS to make the brain stem cells make oligodendrocytes more efficiently? Well, we are in the process of planning and setting up a very small toe-in-the-water clinical trial using bexarotene as a regenerative medicine in MS. Now, this is an area where I have to hand over as an experimental biologist to my clinical colleagues who understand running drug trials and all the gamut of issues associated with that. So this particular trial, which is in large part funded by the MS Society, will be run by my colleague in Cambridge, Dr. Alistair Coles. So Alistair Coles has been engaged in MS clinical trials for many years, is one of the world's experts in this. In fact, Alistair is the one who ran the alemtuzumab trial. So Alistair will be leading this clinical trial, and it'll be a collaboration with Siddharthan Chandran at the University of Edinburgh, and also David Miller, who runs the MS Society-funded imaging centre at the Institute of Neurology in London. So the trial is what is called a randomized placebo-controlled study, and it is addressing the safety and tolerability of bexarotene, an RXR receptor agonist, with the specific objective of testing its ability to promote remyelination. And this particular trial, for reasons I'm happy to discuss later, and as much as I understand it, as I say, I'm not a clinician, will be done in individuals with relapsing remitting MS who are already on beta interferon therapy. Patient treatment recruitment will be in the 2 centres in Cambridge and Edinburgh. The outcomes will be on the basis of MRI scans, which will be done in London, Edinburgh, and the analysis will be done by David Miller's group at London. Now, all being well, the details, the crossing of the T's and dotting of the I's of the funding of this is still under negotiation. But all being well, I am told by Alistair that he's hoping to start recruitment in the autumn of this year, so autumn 2014. A little bit more details on what this trial will involve. Technically, it's called an add-on placebo-controlled trial. Add-on in that individuals will already be on a treatment, beta interferon, and placebo-controlled in that some people will get the drug and some people will get something that looks identical to the drug but isn't, and nobody will know who's got what. Bexarotene will be given in tablet form on a daily basis, and the participants in this first, as I say, toe-in-the-water trial will be individuals with relapsing remitting disease with an EDSS of between 3 and 6, between the ages of 30 and 50. This is a very small-scale trial in the first instance. This is really virgin territory. There is one previous clinical trial with a putative pro-remyelination therapy that has been done anywhere in the world. So this is very early stages. And this initial trial will involve 50 patients, 25 who will get the drug, 25 who will get the placebo. The trial will run over 9 months, 6 months of which will be on treatment. So that's a very optimistic and a very encouraging outcome of the investment in fundamental science. And I think it's worth reiterating here that unless there were the investment by the society in doing the fundamental biological science of identifying this interesting molecule, we'd have never got to the point of being able to do a clinical trial. So I think it illustrates the importance of funding fundamental science for translation. I'm just going to finish on telling you where we're moving with this. It's slightly more complicated than I've told you, and things always are. RXR actually comes in 3 different flavors, an alpha, beta, and a gamma form. The gamma form is the one that is found in the brain. The other types of RXR are found in other tissues in the body. Bexarotene is actually targeting all 3 types of RXR. So although it'll affect the brain stem cells, it'll also affect cells in other tissues. So there will be some what are called off-target or side effects of using bexarotene. They're not expected to be terribly serious and they're expected to be manageable. Nevertheless, there will be some off-target effects. But clearly, it would be much more optimal if we could come up with a drug that was specific to the gamma form of RXR. So it would have optimal and maximal effect on brain stem cells and not affect the other tissue. And that is likely to be a more effective regenerative medicine than bexarotene. So bexarotene we have here and now, and it is absolutely sensible and worth trialing that at this stage. But we're also engaged in what's called a drug development program, where we're going to try and devise new drugs which are specific to the type of RXR that is found in the brain. And that's a much more long-term process. It'll take between 5 and 15 years. So let me just summarize what I've said. Promoting remyelination is important in MS because it'll help restore function, and perhaps even more importantly, it'll stop the nerve fiber loss that is the basis of the progressive phase of the disease. We think in Cambridge and Edinburgh that currently the best way to enhance remyelination is not by cell transplantation, but instead to design drugs which target the stem cells that are already present in the brain. And we've got pretty close to doing that. We're on the brink of trialing just such an approach. The two centres working jointly and with other collaborators in the international community, we've now identified a number of possible ways by which we can come up with regenerative medicines. These are at different stages of development, some at the very early stage, some much more advanced. The most advanced of these is the whole approach around RXR gamma and the bexarotene trial, which will start during the course of this calendar year, all being well. And that I think is where I wanted to conclude, just to emphasize again that we have made, with the help of the investment of the Society, really, I think, better progress than I would have expected when we embarked on this whole venture. When we first started to engage with the MS Society in the notion of setting up a myelin repair center, the idea that we'd have medicines to promote myelin regeneration was really aspirational. And I think as a result of being very fortunate to employ some extremely talented young scientists on MS Society money, we've made much more rapid progress than we might have expected. And I think the notion of regenerative medicines is now a realistic one. And I, for one, am very optimistic about how the next 5 to 10 years is going to unfold. Thank you for attention. I'm happy to answer questions.
Fantastic. Thank you, Robin. Now, officially, we've actually used our time, but I've been given permission to extend a bit so we can have some questions. So if there are some questions, that would be great. And we have one right down at the front in the left.
Very good talk. Thanks, Robin. If remyelination and demyelination, or demyelination and remyelination reflect the relapsing remitting forms of MS, what do you think is happening in the progressive forms when there's no relapsing and remitting phase?
Well, I think that's a very good question, of course. The whole community is trying to get some clarity on. But I mean, there are many components to that answer, but I think it's not unreasonable to think that when you go into the second progressive phase, what's kind of interesting is that people enter the secondary progressive phase at around the same age, regardless of the age of onset of the disease. So this is work from Christian Confrèvre in France that he published a number of years ago, and it's roughly around the age of 40, regardless of when the disease started. And that, to my mind, speaks very eloquently to the idea that what is happening when you enter the secondary progressive phase is you reach an age where the efficiency of remyelination, simply by virtue of your age, has now become so slow that the axons are left naked for longer than is good for them. And when you go into the secondary progressive, you've now entered a phase where actually the new episodes of disease is diminishing, but the axons are remaining exposed, therefore not receiving their nutrient fix from the myelin sheath and undergoing degeneration. And that's an irreversible loss, and the more of those nerve fibers you lose, the more disability you get, so it becomes a progressive disease. And that's how I conceptualize it at the moment.
And another question with Nicola.
Yes, thank you. Thank you, Chairman, and thank you to the speaker. Very clear, I thought. I'm not a scientist, but I thought for a non-scientist, extremely clear. The focus clearly is on repair. I'm just wondering what your work shows and what it helps us on repairing and stopping the damage in the first place.
Well, so these are two separate areas of research. I mean, neither our centre or Edinburgh Centre are really engaged in that whole process of stopping damage, and that, I would say, is probably accounts for 98% plus of the investment in disease treatment interventions over the last 20 or 30 years. So our particular interest has been, our focus is on repair, not the exclusion of those other therapies. And in fact, actually, the success of those therapies is really the basis on which a regenerative medicine is going to work, because clearly, unless you can stop the disease process, then you can regenerate until you're blue in the face and you're still going to have damage. So these two interventions work sequentially with one another.
Okay, two more questions, I think, and then we'll conclude. And I've got one with Sue right at the back, and then one towards the front with Nicola.
Yeah, can I ask, when you talk about the myelin regeneration, so if that becomes what you want it to be, for people who have had the disease for a long time and have entered into that phase where they've had demyelination and nerve loss, regeneration is not going to help them. Is that right, from what I'm reading from what you're saying?
Yeah, that's a very good and very important question. So remyelination clearly won't restore lost nerve fibers. And you can't remyelinate a fiber that isn't there. But at any stage of the disease, you still want to protect what nerve fibers you have. So if one is being very pessimistic about this, then the remyelination therapy will slow the progression. If you're being more optimistic, remyelination therapy will stop the progression. If you're being very optimistic, and in fact there is sufficient biological knowledge to think that this may be the case, if you stop progression of the disease, there may be sufficient what's called plasticity in the brain to actually recover some function. So if you stabilize things, the brain actually is remarkably good at finding new ways of doing old functions that it had lost the ability to do. So it may well be the case that remyelination therapies will lead to a recovery of function by stopping the disease process and allowing the brain to engage in these alternative routes of doing the same function.
Down at the front.
Well, one of the great advantages of medicine in dealing with diseases over the years has been vaccination. Vaccination stimulates the immune system of the body to deal with invasive and problematic things. Do you think the vaccination over the years has been some kind of a cause for the stimulation of the immune system that's affected MS?
Yeah, so this is a good question. Has been investigated pretty thoroughly by immunologists or people who have better immunology knowledge than I have. And it's been one of many things, one of many potential triggers for the disease. And the evidence thus far is there is no compelling evidence thus far that vaccination has been the cause of tipping the immune system into this aberrant form of behavior. So while it's an interesting concept, there's no evidence that that's the case.
Brilliant. So I'm going to make a couple of thank yous, but before I do, I just want to remind people that we do have a science zone right at the back of the hall where you can learn a lot more about myelination, demyelination, and oligodendrocytes and various other things connected to the central nervous system. But my two thank yous, the first is to all of you as people who are part of the MS community that help the MS Society to be able to fund the kind of work that you've heard about today. That's incredibly valuable. And the second, of course, is to Robin Franklin for giving up his Sunday, for coming to talk to us, and for all the work you do day in, day out to make the difference we want you to make. Thank you.